Hello, just let me know whether can you hear my voice or not. There are some issues with this OBS stream, so that's why I think even my image is stuck. Okay. Now OBS stream is not even working. OBS software is not actually working fine. So that's why I think the problem is. Hello. Right now I think it's working fine. Okay. If you are having any query, you can ask in the chat box. Okay. I will look at this live chat stream. If you have any queries, you can ask. Otherwise, we will start directly the lecture. Today's lecture is about the first lecture in this physiology chapter. Okay. So let's start. So first of all, I welcome you to this our new lecture series that is on the second course, that is physiology. There was actually one problem with the last lecture of that cell biology. I will upload it when I get time, because this actually OBS software is not working fine. Even today also I have a lot of trouble to launch this, start this course finally. But let's see, at least we will complete today's part and then we'll move on to the next lecture. So this were further, before that, this was just a short review of the first paid batch. I have given you this 130 page notes also along with the video lectures also. So for the second course, there are also already 49 students have been registered. So if you want to register, you can. And this is the entire course line for the paid batch. Okay. Okay. And most we will just conduct the same lecture on the same day only, as this 9 to 11 is a time of this paid batch. We will have this 11 to 30 to 12 am as a free YouTube lecture series, okay, on the same point. And this would be almost around the entire timeline. I have just specifically taken this genetics, economic, zoology and ecology at the last part, because this ecology will also help you for this prelims also. Okay. So this will be the approx cost. This cost would be for those who are attending each and every module. Like this is only around 9,000. Okay. For those who are attending each and every module. For those who are attending specific module, this is going to be the cost. And trust me, when this entire notes will be ready in the May, at that time the value of this zoology as an optional is obviously going to be increased, because you will be only the optional which is having the point by point notes. Okay. So at that time, this whatever this entire cost is there, this is going to be increased. So I advise you to get the admission as early as possible. Okay. And see, these are the benefits of the entire course. See, as I am giving you the notes exactly by compiling from different sources, you don't need to waste your time in collecting those notes from anywhere. You can just directly read those specific notes. I have given you 130 pages notes of cell biology and 30 hours plus lecture of the cell biology. So you can complete this entire within suppose 5 days only. So in this way, you will just complete almost 95 to 99% of cell biology in the 5 days only. Okay. So as always, I am available for your 24 hours for your queries. We also take Wednesday and Saturday live session at 8 p.m. for the paid batch only. And those who will join all the course, they will get almost 60% discount on the crash course also. The crash course is going to be launched immediately after prelims. Okay. So I will be accompanying you for the entire course duration after prelims only. So I will tell you the how exact time you can give to for the optional and what time you should give for the GS also. So how it is going to be, this course is going to be gone. This is actually for the paid batch. Okay. So I also advise you the same way, just read the lecture and go through the notes if you have. Okay. Now let's start with the first point of today's lecture. That is the composition and the constitutions of blood. In that we will see first the plasma proteins in this today's lecture. So let's see. So whenever you just take the bloods, you can see this 55% or the major part would be this plasma. Okay, it will have all this plasma protein inside, and that would be another component of this blood sample would be that is called as what? Red blood corpuscles and WBCs and platelets and other cellular components will be there. So these are the specific points regarding the blood. That is the overall amount in the blood is around 5 to 6 liter. Specific gravity is around 1.05 and viscosity is 4 to 5 times that of water. You can just exclude this point if you want, not much important. pH, that is 7.4, it is alkaline, that is important point. So the composition of blood mainly just categorized into two, plasma and cells. We will see this plasma today. So let's see the plasma first. So in this plasma also, this is actually the clear straw colored fluid portion of this entire blood. Apart from this cellular component, it contains this, most of the part is 91% is water, and out of this 9% solids are there. In between that 1% is inorganic molecules and these 8% are organic molecules. Okay. So let's see what are these 8% of organic molecules contain. See, these are the 7% are plasma proteins. Okay. So proteins are present, and 1% is non-protein nitrogenous components like this urea, creatinine, creatine, all are present. So now our main focus is this 7% of plasma protein. So let's see what those 7% of plasma proteins mainly are. See, out of these main components are like this pre-albumin, albumin, globulins, fibrinogen, prothrombin, and these are their functions. Like this pre-albumin binds to this T4, T3 and vitamin A. Okay, and also transport them. This albumin, this controls this colloidal osmotic pressure, means COP. Okay, it contains the filtration rate. Okay, inside the blood, this albumin. Then there is the globulin. See what are these functions of these globulins are like this, inhibiting the certain blood proteins or the enzymes in the blood, and they are also responsible for the natural and the acquired immunity. And these two, fibrinogen and prothrombin. Fibrinogen is and prothrombin, both of them help in blood clotting. Out of this, fibrinogen is precursor of fibrin and prothrombin is a precursor of thrombin. Then these are the different forms of globulins are there, like this in case of, and the functions like glycoproteins for the immunity and physiological functions. In the lipoprotein mainly, three types of lipoproteins are there: HDL, LDL and VLDL. See, these high density lipoproteins are also referred to as good cholesterol. Why they are called as good cholesterol? Because their density is low, so their size is also low. Because of their smaller size, they can easily pass throughout this entire blood capillary or blood arteries. So they are not very less tend to form a clog, okay, clog or plaque of this fat or proteins or lipid. But in case of this very low density lipoprotein is that as their density is very low, their size is very big. So as their size is very big, they can form a plaque or they can just deposit it on this artery. So they form a plaque and they just block this entire artery. So that's why they are called as a bad cholesterol. Okay. So these are the two are bad cholesterol. This HDL is what? Good cholesterol. This transferring globulin is what? Transporting this and iron absorption and transport in the gastrointestinal tract. Immunoglobulins or gamma globulin is about the specific globulins. Now let's see specific functions of the plasma proteins. The first is, I have mentioned you that this is also helpful in the coagulation of blood. The next, that is the helps in maintaining the colloidal osmotic pressure. Now let's see how these plasma proteins are helping maintaining the colloidal osmotic pressure. See, the thing is that this is, you can say these are the two parts. Okay, two parts of this entire diagram are there. Okay, this part is what? Arteriolar end towards the artery, and this part is venous end towards the vein. So what happens is that how these plasma proteins are contributing this colloidal osmotic pressure is that, see, this there is fluid in this artery or there is fluid in this vein, right? So that fluid is exerting some pressure. Okay, and surrounding it at this entire capillary or artery are having pores around it. So they are exchanging fluids with the surrounding interstitial fluid. What is the interstitial fluid? It is fluid in between the cells, you can say, or surrounding fluid around this entire capillary network or this venous network is there. That is called as what? Interstitial fluid. So this blood inside this is continuously exchanging or just filtrating things in between this interstitial fluid and blood. So there is for that filtration there is a force that is acts, and that force is called as what? Pressure gradient. Okay. In case of this arteriolar end, that pressure gradient is provided like this. Hydrostatic pressure means what? Water pressure, and this COP means what? Colloidal osmotic pressure. That is because of what? Because of the plasma proteins. Okay. The constituents of this plasma proteins at this side is same, at this side is also same, but hydrostatic pressure is higher at this arteriolar side. So what will happen? Hydrostatic pressure is around 37 of mmHg and COP is around 12 mmHg. So the effective pressure is how much? The COP is around what? 25 mmHg. So effective filtration rate is what? 12 mmHg. The hydrostatic pressure minus COP. The pressure by water minus the pressure by proteins. But after the filter, so that's why filtration is occurring. Okay, so that's why filtration is occurring. So the blood is going to filter, water will come out. Now what will happen? The hydrostatic pressure in the venous end will decrease because we have filtrated a lot of part in this first part only. So here the hydrostatic pressure is less. That is, you can see it is 15 mmHg only. Earlier it was 37 mmHg. Now as the hydrostatic pressure has been decreased to 15, but the COP is same, the proteins are going to be same inside the blood. So the COP pressure is going to be 25 mmHg only. So now the pressure gradient, the difference in between this 25 mmHg and 15 mmHg is what? 10. Now with that pressure what will happen? This absorption is happening. Now you can see this almost 12 and 10 are along equal. So this body maintain a balance in between this filtration rate and absorption rate. But what will happen sometimes is that if this protein or the plasma proteins concentration changes, so at that time this COP value will get changed. Okay. If the plasma proteins are changed, suppose this COP now becomes only 5 mmHg. Now you can see the 37 minus 5 will be what? 32 mmHg. So the filtration will be occur with the 32 mmHg, and here you can see the 5 minus 15, it will be not even give any false result. So that just means what? It is this 32 mmHg and the pressure absorption will be very less. Okay. So because of that very less absorption, what will happen? Whatever this filtration or whatever the water that was earlier just gone out from this artery, that water will not taken out through this absorption. So that water will accumulate somewhere. Okay. That water will accumulate on this interstitial tissue, and when such water accumulates in the interstitial fluid, at that time that accumulation is referred to as what? Edema. Okay. And why this is edema is occurring? Because now filtration rate is too high and absorption rate is too less. So that's why the body cannot absorb whatever the water it has earlier lost. So that's why it is accumulating the water outside this entire blood capillary network. So in where it actually accumulates? In the interstitial fluid. So that is referred to as what? Edema. So this is now this COP, this colloidal osmotic pressure, which is actually you can see just controlling this entire you can say this. Now all of these forces which are there, they are referred to as what? These Starling forces. Okay. So now this COP is actually doing what? It is just regulating this entire Starling forces. So that's why it is important. The next is acid-base balance. Now let's see how these plasma proteins are maintaining the acid-base balance. So the thing is that the protein which are present in the blood, they are having this N-terminal end, C-terminal end. The N-terminal end adds physiological pH, that is 7.4, present in the form of this NH3 plus, and C-terminal present in the form of this COO minus. So how this protein will play a role of buffer, or how this acid-base balance is going to be occurred with the help of protein is that even if you add a small amount of base to this blood, base means what? OH minus. So this small amount of base or OH minus ions will get adjusted with the help of this NH3 plus N or N-terminal end. So they will be balanced. So that why there will be no effect on pH. So that's why you can see now acid-base balance is there. Base balance is there. Now let's see how acid is balanced. So when small amount of acid is added, at that time this H plus of this added acid will be stabilized by the COO minus, that is C terminal of the protein. Okay. Now in this way we can see acid and base are now both balanced because of what? Because of this plasma protein present inside the blood. Okay. So that was the thing with this plasma protein is there. The more detailed lectures are there in the paid batch. If you want to join, I have already just completed the first lecture. You can still join this entire physiology course. If you want to join, you can just contact me directly on the Telegram, or you can go to my website, that is www.shivaschool.in. So there you can just place your order and make a payment. So till then, have a nice day. Bye.